The presented protocol describes a Neurite Outgrowth assay that is highly relevant to human biology and neurotoxicity assessment of small molecule compounds. High translational potential and physiological relevance of human neural progenitor cells as a primary human cell model offer a considerable advantage in Neurite outgrowth-related drug discovery screening and neurotoxicity assessment.
Utilizing the presented protocol, human-induced pluripotent stem cell and human embryonic stem cell-derived neurons can also be used as two alternative cell sources for the Neurite Outgrowth assay and neurotoxicity assessment.
Begin by collecting the media with floating neurospheres and transferring it to a 50 milliliter conical tube. Centrifuge the tube at 300 to 400 times G for 3 minutes. Then carefully aspirate the supernatant and resuspend the spheres in 500 microliters of defrosted cell dissociation reagent. Incubate the spheres at 37 degrees Celsius for 5 to 15 minutes. Then add 5 to 10 milliliters of pre-warmed culture media and centrifuge the tube at 300 to 400 times G for 5 minutes to sediment the neurospheres. Aspirate the supernatant and add 2 milliliters of culture media, pipetting up and down with a 1000 microliter pipette until the neurospheres form a single cell suspension. After counting the cells, add 2 to 3 million cells to a T25 flask in 10 milliliters of culture media. Replace half the culture media every 3 days.